Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Adina Fahim, and today I'm going to be presenting my science fair project, Effects of Different Water Types on Plant Growth at Different Stages. Water is critical for the germination of seeds and zkwas as a carrier for mineral nutrients essential for growth. Different water types influence growth, including tap water, distilled water, rainwater, softened water, hard water, and plants with sugar and salt. Water with mineral content formed when it passes to deposit of gypsum and limestone containing calcium, magnesium, and carbonates in hard water. Hard water does not influence plant growth. However, softening of hard water by removing minerals can compromise plant growth. Presence of NaCl in soil and water roots, including all NaCl ions, which result in regulation and lost energy of plants. This stresses the plant and compromises growth and development. Soft water treatments are frequently suggested to improve the uptake of required nutrients for the soil transported through the roots. Rationale: different water types are used in communities and households for watering of domestic plants, including soft water, rainwater, hard water, and others. As different water types are affected plant growth, practice of water softening in households are common, and plant watering with the same water is usually practiced. This could compromise the domestic plant growth and domestic level farming processes common in our communities. Does the use of different water types, hard water, soft water, and mineral water, influence different stages of domestic plant growth? Hypothesis and aim. The use of different water types includes hard water, HW, soft water, HW, and mineral water, MW, will significantly influence the domestic plant growth at different stages. It is also hypothesized that plants are at different stages in presence of HW will show significantly better growth as compared to SW with low mineral content. The aim of the present study was to investigate the influence of water type HW, SW, and MW on the growth of plants at different stages. Experimental design and practice. Experimental design include Group 1. Plants and seeds watered with hard water. Group 2. Plants and seeds watered with domestically softened water. Group 3. Plants and seeds watered with bottled mineral water. Studied assessments were cotton formation. 5 grass seeds were planted in 3 different pots, which were watered with HW, SW, and MW. The duration in hours from the time of planting to the time of germination was recorded. Percentage of seed germination. Grass seeds were planted, 5 in each pot, in 3 pots. Pots were watered and the percentage of seed germination was calculated for each group. Plant height assessment. ZZ plants of smaller heights were watered with respective water types for 30 days. The length of the plant were measured from the borders of the container to the top of the plant. Dry weight of plant. ZZ plant was removed from the soil after 30 days of watering, and they were assessed for dry weight. Plants were placed in an oven set at 100 degrees Fahrenheit overnight. The plants were placed in Ziploc plastic bags until cooled. Plants were measured for weight on a detailed scale for day weight. Means and standard deviation were compared among the groups using statistics tests. A p-value of less than 0.05 was considered statistically significant. This contains the data for germination duration and germination percentage for the grass seeds. Figure 1. Seed germination in HWA, HWB, and MWC. This table contains the data for the plant height of plant ZZ. Figure 2. Plant growth and height in HWA, HWB, and MWC. This table contains the data of plant dry weight of the plant ZZ. Figure 3. Dry plant weight assessment for HWA, HWB, and MWC. Interpretation and discussion. HW shows better plant growth. Seed germination duration was longer, and germination percentage was high, as compared to HW and MW samples. Therefore, the hypothesis was accepted. Water softening process causes the removal of high quantities of Mg and Ca. Ca in plant growth is required in high concentration for plant cell wall and cell membrane. Ca is only extremely member, and once it is placed in plant location, it cannot be mobilized to other plant areas. Therefore, a continuous supply of high amount of Ca is needed for optimum plant growth. One of the common symptoms of Ca deficiency in soil is death of growing plant, premature shedding, and end root. Mg is also reduced in water softening processes. Mg forms the core of the chlorophyll membrane. In plants, associated with enzyme cellular of plant metabolism. metabolism. Any deficiency of Mg can cause sudden plant growth due to compromise in the cellular biochemical. Lack of Mg causes change in plant color from green to yellow due to storage of chlorophyll in plant leaves. It was observed that germination of seeds is increased by increasing the Ca content. However, it is no effect on However, this is effect is on some and not all plant species in their experiment. Ca metabolism in plants vary, and therefore plant may react differently to lack of Ca and Mg levels, specifically in presence of Sw. Exposure to different water types was performed for 30 days only. However, experimenting with different plant species and longer exposure to different water types would show a more comprehensive outcome of water types and plant growth. Conclusion. Within the limits of this experiment, seeds watered with hard water showed faster germination and higher germination percentage than seeds watered with soft water and mineral water. Hard water plants showed greater plant growth and plant height and dry weight compared to Sw and Mw plants for 30 days. Bibliography.